All right, a little mini video on genus, species, and strain. We'll have that at the very bottom. Remember, scientific names are in a two-name format, which we call binomial nomenclature. The genus is going to be the first name. Rules for the genus will be that these are capitalized versus the species is going to be lowercase. The other rule for these names is they will always be underlined or in italics. Underlined or italics. And that is just our scientific naming strategy. Okay, some examples. Genus and species such as Staphylococcus Aureus and Staphylococcus Aureus. Staphylococcus would be the genus, Aureus would be the species. This is the staph that we think of with pathogenic staph infections, and you'll see the essay if you possibly run into a strain that is resistant to methicillin. You may have heard of MRSA, and MRSA would just be a methicillin-resistant strain of Staphylococcus. There are hundreds of different strains of Staphylococcus aureus and hundreds also of different types of MRSA strains. So there can be very slight differences that impact organisms, allowing them to grow in certain conditions better than others. And for patient care, that can impact treatment of antibiotics. Some antibiotics may work better on one strain versus another strain. Another example of genus and species is going to be Escherichia coli. I'll underline it or put it in italics, and you commonly know this organism as E. coli. So this also gives you an idea that we can abbreviate the genus name as the capital of the first letter, E, and then coli for that species name. The naming strategy for all of these organisms, whoever identifies it gets naming rights. The only names I have not seen have been profane in origin, but looking at these are a couple of good examples. Staphylococcus. Staph refers to cluster, and coccus is going to refer to berries. So this is the microscopic morphology of staph under the microscope. So Staphylococcus under the microscope would look like a cluster of berries, small circles. The aureus refers to its coloration, gold. However, that's when it's on a solid medium and able to form those colonies, because we won't see the color of that cell as gold until it has been able to multiply into a colony. As for E. coli, Dr. Escherick was the lab physician, and this particular bacteria was found in the colon. So this bacteria was found in Dr. Escherick's patient's colon. All right, there are a couple of examples for you. Many, many more in the PowerPoint notes.